11.4 Part 1 Meiosis. As geneticists in the early 1900s applied Mendel's laws, they wondered where genes might be located. They expected genes to be carried on structures inside the cells, but which structures? What cellular processes could account for segregation and independent assortment, as Mendel has described? How many sets of genes do multicellular organisms inherit? The diploid cells of most adult organisms contain 2 complete sets of inherited chromosomes and 2 complete sets of genes. Chromosome number. Chromosomes, those strands of DNA and proteins inside the cell nucleus, are the carriers of genes. The genes are located in specific positions on the chromosomes. A body cell in an adult fruit fly has 8 chromosomes, as shown in the figure. 4 of the chromosomes come from its male parent, and 4 come from its female parent. These 2 sets of chromosomes are homologous, meaning that each of the 4 chromosomes from the male parent has a corresponding chromosome from the female parent. A cell that contains both sets of homologous chromosomes is a diploid, meaning 2 sets. The diploid number of chromosomes is sometimes represented by the symbol 2n. For the fruit fly, the diploid number is 8, which can be written as 2n equals 8, where n represents twice the number of chromosomes in a sperm or egg cell. Some cells contain only a single set of chromosomes, and therefore a single set of genes. Such cells are haploid, meaning 1 set. The gametes of sexually reproducing organisms are haploid. For a fruit fly gamete, the haploid number is 4, which can be written as n equals 4. What event occurs during each phase of meiosis? In prophase 1 of meiosis, each replicated chromosome pair with its corresponding homologous chromosome. During metaphase 1 of meiosis, paired homologous chromosomes line up across the center of the cell. During anaphase 1, spindle fibers pull each homologous chromosome pair toward opposite ends of the cell. In telophase 1, a nuclear membrane forms around each cluster of chromosomes. Cytokinesis follows telophase 1, forming 2 new cells. What events occur during each phase of meiosis? As the cell enters prophase 2, their chromosomes, each consisting of 2 chromatids, becomes visible. The final 4 phases of meiosis 2 are similar to those in meiosis 1. However, the result is 4 haploid daughter cells. Meiosis is a process in which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in a diploid cell. Meiosis usually involves 2 distinct divisions called meiosis 1 and meiosis 2. By the end of meiosis 2, the diploid cells become 4 haploid cells. Just prior to meiosis 1, the cell undergoes a round of chromosome replication called interphase 1. Each replicated chromosome consists of 2 identical chromatids joined at the center. The haploid cells produced by meiosis 2 are gametes. In male animals, these gametes are called sperm. In some plants, pollen grains contain haploid sperm cells. In female animals, generally only one of the cells produced by meiosis is involved in reproduction. The female gamete is called an egg in an animal and an egg cell in some plants. Fertilization, the fusion of male and female gametes, generates new combinations of alleles in a zygote. The zygote undergoes cell division by mitosis and eventually forms a new organism. How is meiosis different from mitosis? In mitosis, when the 2 sets of genetic material separate, each daughter cell receives 1 complete set of chromosomes. In meiosis, homologous chromosomes line up and then move to separate daughter cells. How is meiosis different from mitosis? Mitosis does not normally change the chromosome number of the original cell. This is not the case for meiosis, which reduces the chromosome number by half. Mitosis results in the production of two genetically identical diploid cells, whereas meiosis produces four genetically different haploid cells. Mitosis is a form of asexual reproduction, whereas meiosis is an early step in sexual reproduction. There are three other ways in which these two processes differ. Replication and separation of genetic materials. In mitosis, when the two sets of genetic materials separate, each daughter cell receives one complete set of chromosomes. In meiosis, homologous chromosomes line up and then move to separate daughter cells. As a result, the two alleles for each gene segregate from each other and end up in different cells. The sorting and recombining of genes in meiosis results in a greater variety of possible gene combinations that could result from mitosis. Mitosis does not normally change the chromosome number of the original cell. Meiosis reduces the chromosome number by half. A diploid cell that enters mitosis with 8 chromosomes will divide to produce two diploid daughter cells, each of which also has 8 chromosomes. On the other hand, a diploid cell that enters meiosis with 8 chromosomes will pass through two mitotic divisions to produce 4 haploid gamete cells, each with only 4 chromosomes. Mitosis is a single-cell division resulting in the production of two genetically identical diploid daughter cells. Meiosis requires 2 rounds of cell division and, in most organisms, produces a total of 4 genetically different haploid daughter cells. How can two alleles from different genes be inherited together? Alleles of different genes tend to be inherited together from one generation to the next when those genes are located on the same chromosome. Thomas Hunt Morgan's research on fruit flies led him to the principle of gene linkage. After identifying more than 50 Drosophila fruit fly genes, Morgan discovered that many of them appeared to be linked together in ways that seemed to violate the principle of independent assortment. For example, Morgan used a fly with reddish-orange eyes and miniature wings in a series of test crosses. His results showed that the genes for those two traits were almost always inherited together. Only rarely did the genes separate from each other. Morgan's findings led to 2 remarkable conclusions. First, each chromosome is actually a group of linked genes. Secondly, it is the chromosome that assorts independently, not individual genes. Alleles of different genes tend to be inherited together when those genes are located on the same chromosome.